this is an all-in-one video, so I'm going to go through the whole of the genetics topic. We'll be looking at monohybrid crosses, dihybrid crosses, sex linkage, epistasis, pedigree diagrams, so it's going to be a real beast of a video. To start with, we need to go over genetics basics though, and in order to do that we'll be looking initially at monohybrid crosses, which you should hopefully have met at GCSE and IGCSE. Before we dive into that, let's remind ourselves of some key definitions. So remember that a gene is a section of DNA which codes for a protein. An allele is a different form of the same gene. So for example, if you were looking at eye colour, that could be uppercase B for brown eyes, and then you could have an allele which is lowercase b for blue eyes. Remember, some alleles are dominant, and that means that the allele is expressed even when there's only one copy of the particular allele. Now looking at a recessive allele, this is only expressed if the dominant allele is absent. The phenotype is the actual appearance of an organism, e.g. having blue eyes. The genotype is the alleles an organism has. A homozygous individual is the same as a pure breeding individual, which means that both alleles are the same. A heterozygous individual has two different alleles. What I want to do now is jump in with a monohybrid example. So we're going to cross a heterozygous brown-eyed woman with a homozygous blue-eyed man. So what we're having a look at is what the outcome will be in terms of their offspring. So what is the chance of them having blue-eyed kids, brown-eyed kids, etc. So always lay it out like this. So first of all, your phenotype, which remember is the actual appearance. So we know that the man has blue eyes and the woman has brown eyes. The genotype is the alleles that the individual has. So we're going to use the letter B. Because the woman we've been told is heterozygous, it means that she contains a big B, small b. The man is homozygous, which means, first of all, that both the alleles are the same, and because blue eyes are a recessive trait, that means they're both lowercase b. Now we need to determine the gametes, so the sex cells, which remember are the sperm and the eggs. Because of meiosis, it means that we have a haploid number of chromosomes, so the man's sperm are either lowercase b or lowercase b, the woman's eggs are either uppercase b or lowercase b, and now we're ready for the all-important Punnett square. Doesn't matter which way around you do this, but let's add those gametes. So there's the man's sperm, here's the woman's eggs. So this offspring will be big B, small b, this offspring will be the same, this offspring will be homozygous recessive, and the same with this offspring. So now whatever you do, make sure you link your outcomes phenotype with their genotype, so we can see that both of these offspring will have brown eyes. So 50%, or 1 in 2, or 0.5, these numbers all mean the same, will be big B, small b, which means they'll have brown eyes. And then 50% will have blue eyes. And now we're going to take all the other different parts of the genetic topic in turn. Just so you know, this really useful PowerPoint which I'm using to aid my explanation was provided by Tony Graham from the TES website. So I'm just making sure I'm acknowledging that here. So dihybrid crosses are those where we consider the inheritance of two characteristics at the same time. This makes sense because di means two, so that's why we're looking at two characteristics being inherited at the same time. Here the two different genes are inherited on two different chromosomes. The reason we're pointing that out is because we need to acknowledge that these genes are unlinked, and that will make a real difference to the gametes which are produced. Now hopefully you've met Gregor Mendel before. He was a monk who was basically the forefather of inheritance. He studied peas, so Mendel investigated the inheritance of two characteristics of pea plants at the same time. He looked firstly at seed shape, where the round shape is dominant to the wrinkled shape, and secondly he looked at the seed colour, noticed that yellow coloured seeds were dominant to green. And so if we look at the four possible combinations of these two characteristics, we notice therefore that peas can be round and yellow, round in green, wrinkled in yellow, and wrinkled in green. Mendel carried out a cross between two pure breeding plants. This therefore means that these plants are homozygous because that's what pure breeding means, which means they have two of the same allele. And the two pure breeding plants he crossed was round yellow seeds and wrinkled green seeds. So let's have a look at that first cross. It's been really handy that we've got the pictures here so you can actually see them. There's our round yellow phenotype. Here's our wrinkled green phenotype. Because we were previously told that round was dominant, and as was the colour yellow, this explains therefore why they're given by the uppercase version of their letters. We were told that the wrinkled green versions of those seeds were recessive, and that explains why we're seeing the lowercase version here. And therefore we know that all the gametes produced by the round yellow seeds will be capital R, capital Y. Remember, the gametes are the sex cells. And produced from the wrinkled green seed, we know that all those gametes will be lowercase r, lowercase y. And so if we were to do a cross over here, we can see that all our seeds would have this genotype, as indeed they do, and that obviously corresponds to that round yellow seed. So all the F1 generation would be heterozygous, meaning that they have different alleles for both characteristics, meaning that they would all be round and yellow. And then, because it's very similar to what Mendel did with the monohybrid crosses, he then crossed two of the F1 generation together. So we're taking two of these seeds, you can see them here, and we're crossing them. So we know again that their phenotype will be round yellow, but their genotype, as we've already said, was that heterozygous genotype, meaning that you've got a huge combination of gametes being produced. And that's the major difference here when you're looking at dihybrid crosses. These alleles can rearrange themselves in any way, so that's why we've got capital R, capital Y here, capital R, lowercase y here, lowercase r, capital Y here, lowercase r, lowercase y here. And that's the same for both seeds. And so when we do our cross, we need a giant Punnett square for this, which we've got here, and handily we've been shown what that phenotype would be. So we've got our dominant phenotype here with our round yellow seeds. The same is true here, here and here. The reason it's gone green at this point is because we have that homozygous recessive genotype. And as we look down, there'll actually only be one seed which is green and wrinkled, and that's because it has the homozygous recessive genotype. So if we were to look at our ratios, you literally just count them from the table. How many round yellow seeds do we have? 1, 2, 3, 4, 5, 6, 7, 8, 9. And here they are. How many round greens? 1, 2, 3. How many wrinkled yellows? 1, 2, 3. And finally that one wrinkled green seed. And as you can see, we have done a ratio. We've pointed out how those phenotypes link to the ratio. And notice this 9:3 to 3:1 is the typical ratio expected in a dihybrid cross. So if you're ever doing a dihybrid cross with unlinked genes, which involve that heterozygous genotype, you will find a 9:3 to 3:1 ratio. So first of all, let's start by defining the term multiple alleles. And this is when a gene has more than two alleles. And a good example of this is human ABO blood groups. Now there are three alleles associated with the gene I, which stands for the immunoglobulin gene. And this leads to the presence of different antigens on the cell surface membrane of red blood cells. And so this is the really important bit. Allele IA leads to the production of antigen A. Allele IB leads to the production of antigen B. And allele O, which actually doesn't lead to the production of either antigen. So there's no A or B produced. Now, despite the fact that there are three different antigens, only two can be present in an individual at any one time. So only two alleles can be present in an individual. And seriously, the most important thing to take away from this video is that the alleles A and B are codominant, whereas O is recessive to both IA and IB, and that's going to be so important when we look at some crosses. So first of all, pointing out the variety of different blood groups which exist. Because of antigen A, you've got the blood group A. Because of antigen B, you have blood group B. Because A and B are codominant, it means that you have a blood group AB. And then finally, you have the blood group O. So what's the potential genotype for blood group A? Well, clearly you could have that homozygous version, or because that O allele is recessive, effectively if you have only one copy of that A, you'll still have that A blood group. The same is true for blood group B. You could have the homozygous version, or you could have the version which is combined with that O allele. AB, remember, A and B are codominant, so this is the only potential genotype. And because O is recessive, the only potential genotype for that final blood group is IO-IO. So let's look at some test crosses. Cross a mother who is heterozygous for blood group A with a father who is heterozygous for blood group B. So starting with the mother's phenotype, we know that she is blood group A, the father has a blood group B. What's their genotype? Well, because they're heterozygous, we know that they must contain that O allele. What are the potential gametes? Well, the mother's eggs are either IA or IO. The father's sperm are either IB or IO. Ready for that Punnett square now? So we've got the father and the mother. Let's copy in those gamete genotypes. Let's do the cross. Doesn't matter which way around you draw the alleles, by the way. So we have quite an interesting scenario here, because if we have a look at each of the offspring genotypes, let's start with this offspring. Because that A and B allele are codominant, then we know that this offspring will have an AB blood group. This offspring here will just be an A blood group, because remember that A is recessive to O. This offspring is very similar, but it's this time the B blood group. And then finally, this offspring is an O blood group. And I've pointed out all my genotypes and how they link with my phenotypes. So 1 to 1 to 1 is your ratio. The first thing to point out is that it is a recessive disease, which means it's only displayed in the phenotype when the dominant allele is absent. And that's going to be incredibly important later on, and I'll point out why that is. Sickle cell anemia means that rather than getting your normal biconcave red blood cells, so hopefully you remember they're quite donut in shape, and this is the normal situation when they have a large surface area to volume ratio, which means they can transport lots of oxygen. Unfortunately, in the case of sickle cell anemia, you find that the red blood cells are sickle shaped, which kind of means they're like crescent moons. And because of this, far less oxygen is transported, and that's due to a smaller surface area to volume ratio, so less oxygen transported, and you do find that there's quite a lot of internal bleeding. So the mortality rate for this disease is quite high. Because this video is all to do with how it's inherited, I now need to point out to you that sickle cell anemia is only carried on the X chromosome. But why is that so important? And that's because if you look at the sex chromosomes of a female, remember that she will have 2 X's, and a man will have an X and a Y. So if we say that sickle cell anemia is only carried on the X chromosome, then it can only be carried on these chromosomes. It cannot be carried on the Y chromosome. So if we look at the combination of alleles which result in sickle cell anemia, let's have a look now. So here's one female, and you're going to use the letter H to represent sickle cell anemia, and I'm using the lowercase h because it's a recessive disease, as I've already mentioned, and I'm going to use the uppercase H to show someone who is healthy. So if we have a look at the combination of alleles, this person here will be healthy, and remember they are homozygous because they have 2 copies of that uppercase H. This person will also be healthy, and that's because sickle cell anemia is a recessive disease. We can also see that they are heterozygous because they have 1 uppercase H and 1 lowercase. The final combination for females is 2 lowercase h's. This person unfortunately has sickle cell anemia because they have 2 copies of that recessive allele. And all the time we're very interested in what's going on here, here and here. If we now look at the potential male combinations of alleles, as we've already said, males contain the sex chromosomes XY. So what about if we were to do an uppercase H? I don't write anything here because remember I've already said that only the X chromosomes carries the genes for anemia. So this person will be male and they will be healthy. The only other combination is this setup, which again is a male, but unfortunately they have sickle cell anemia. So the things I really want to point out to you at this point is, remember that sickle cell anemia is carried by that X chromosome. You must remember that it is a recessive disease and that it's a disease of the blood which lowers the oxygen carrying capacities. Now we're ready to do some genetic crosses to work out the chances of a child having sickle cell anemia. So in cross 1, we're going to have a healthy male and carrier female. What is the probability of their child having sickle cell anemia? So the important thing is to work out what's going on with our genotype. First of all, we know we have a male, so they're going to be XY. We have a female, so they're going to be XX. The male is healthy, which means he must have an uppercase allele, because that would mean that he does not have the disease. Now the carrier female is also healthy, so she's going to have 1 uppercase H and 1 lowercase h. So we're now ready to lay out our answer properly. I always use the same method, so we're going to say male and female. Use this template always when answering these questions. So the phenotype, what is that physical appearance? So the male's healthy and the female's a carrier. The genotype now is the alleles contained by the organism, so we've already written that to make it easier for ourselves. We've got XY with that capital H there, XX, 1 large H, 1 small h. Next up, the gametes. Remember, those are the sex cells, so some of those sperm will have the XH, they'll be female and they'll be healthy sperm, and some will have that Y chromosome, which means that they'll be male sperm. The female, some of her eggs will be healthy and some will carry that gene for anemia. And if you don't like what I'm doing here, you must watch my other videos on Punnett squares, because this is assuming that you're okay with GCSE level Punnett square theory. So we're ready to do our Punnett square now. Our male, we just copy down the gametes that we've worked out. So we have that X and that large H, the Y. The female, we have X, uppercase H, X, lowercase h. Now we're ready to cross those alleles, so you just rewrite them. And now you need to point out, and this is what people always forget to do, what these genotypes are showing. So let's look at this child here. What's going on there? Well, we have a female, so 25% will be female and they will be healthy, and that's because they have that combination of big H, big H. If we look at this one now, another 25% will be female. They'll also be healthy, but they'll be carriers of sickle cell anemia. And we'll point out that genotype, it's because they have that combination of the big H, small h. Looking at this male, 25% will be male and they will be healthy, and that's because they have that combination of big H and then that Y chromosome. And then unfortunately for this male down here, we have 25% will be male with sickle cell anemia, and if we write that genotype, you can see that it's small h, Y. Let's just point out a few key things though. So first of all, a pedigree chart shows the inheritance of a particular trait within a family, and it usually tracks a particular trait through several generations. And so really you have a family tree. Notice that a square always represents a male individual and a circle represents a female. If you have that solid filled in shape, then it means that the particular individual has the trait. If it's empty or unshaded, it means that they do not have the trait. The straight horizontal line between the male and the female represents a mating line. So we're talking about this line here or here. And then a vertical line descending from that line represents the offspring. So we can see in this first example that they have 4 children. 2 of those children are male, 2 are female, and notice that the mother up here and the firstborn son over here have the particular trait. It's important to notice that the pedigree only shows the phenotype, not the genotype, so we're looking at the trait. But now I've talked you through some key notes. I now want to take you through an example. And it says that people with night blindness have difficulty seeing in dim light. The allele for night blindness is dominant. That's so important to the allele for normal vision, which is lowercase n, and these alleles are not carried on the sex chromosomes. So that's another important thing to notice. Let's have a look at what we have. So here's our affected female. They have night blindness. Just going to make a note here. Here's the female. They have night blindness. And remember that that night blindness is caused by that dominant allele. Here's the male who's unaffected. They have 1, 2, 3, 4, 5, 6, 7 children. These 2 are sons, then they have 3 daughters, another son, and finally another daughter. And the shaded ones show affected individuals with night blindness. So individual 12 is a boy. What is his phenotype? So make sure you remember that the phenotype is the physical appearance. We're looking at this individual here. Because we know that night blindness is caused by a dominant allele, it makes sense therefore that both of these must be, meaning that there's no way that individual 12 can have night blindness. So we're going to say that they have normal vision, because remember we're looking at the physical appearance. What is the genotype of individual 1? Explain the evidence for your answer. So we're looking at this individual here. We're looking for a genotype, so we need a combination of alleles. I've already pointed out that they must have a capital N in order to have night blindness. The question is now, does individual 1 have the homozygous dominant genotype or the heterozygous version of that genotype? And the way to work that out is by looking at all the offspring. Obviously, if they had this genotype, then every single one of the offspring would have night blindness. This isn't the case, which stands to reason, therefore, that they are heterozygous. So let's write a reason for that. So she must have a capital N in order to have night blindness, and also a lowercase n because some of her children have normal vision. So make sure you write that down, because I'm now going to rub it out so we can write the third answer nice and easily without getting muddled. So what is the probability that the next child born to individuals 10 and 11 will be a girl with night blindness? Show your working. So 2 things to point out. First of all, that they have to be a girl, and second of all, that they have night blindness. So we're looking at individuals 10 and 11. So in order to work this out, we need to actually do a Punnett square. Because we're looking at individual 10, which is a female, we know that they have 2 X chromosomes. In terms of referring to the night blindness aspect, we know that they're heterozygous because the father can't have passed on a dominant allele, because otherwise they would have night blindness too. Looking at the male individual now, their sex chromosomes are XY. We know that they're unaffected, so therefore they must be homozygous recessive. So now we can do a Punnett square. So there's the mother. There's the father. Let's do that cross. And then now let's match up these genotypes with the phenotypes. So we know that this individual will be a girl with night blindness. This individual will be a girl with normal vision. This offspring will be a boy with night blindness. And then lastly, this individual will be a boy with normal vision. So if we answer the question, what is the probability that the next girl born to individuals 10 and 11 will be a girl with night blindness? It's 25%. or 0.25 if we're going to be official and refer to probability. Just to recap with dihybrid crosses, remember we were looking at two different genes inherited on two different chromosomes, and fundamentally that is the biggest difference when we look at autosomal linkage versus the dihybrid cross. We're looking now at two different genes which are inherited on the same chromosome. Just to remind you that an autosome is a non-sex chromosome, which makes sense. Autosomal linkage is all about looking at two different genes located on the same chromosome, which happens to not be a sex chromosome. Sorry, this is quite wordy, but it is important that you know it. So if genes are located on the same chromosome, they are often inherited together. And if you're asked to provide a definition of autosomal linkage, that is indeed the definition you should provide. So for example, if the alleles capital A, capital B are found on one chromosome and lowercase a and lowercase b alleles are found on the other chromosome, the person will either inherit capital A, capital B or lowercase a, lowercase b. Whereas do you remember when we looked at the dihybrid cross, it was possible to have these four different combinations of gametes. And notice that we're assuming that no crossing over has occurred during prophase 1 of meiosis, which remember is when portions of those chromosomes swap places to increase genetic variation. And as always, it will become a lot more straightforward if we look at a key example. So in fruit flies, genes for body colour and wing length are autosomal linked traits. Notice that capital G is the allele for a grey body, lowercase g is the allele for a black body, capital N produces long wings, and lowercase n produces short, otherwise known as vestigial wings. In this situation, we're being asked to cross a purebred grey longed wing fly with a purebred black short-winged fly. Let's work out the genotypes quickly. Remember, if they're purebred, we're looking at a homozygous situation where those alleles are the same. We have our key above, so we know that we've got a grey fly, so we need a capital G. It's pure breeding, so we've got two versions of that allele. It's longed winged, so we know that that's capital N, again purebred, so it's another capital N. We're crossing it with a purebred black short-winged fly, and so using our key above, we know that this is the genotype for that second fly. So let's look down, look how I've arranged it. We've got our male and our female with its phenotype, which remember is the physical appearance. The genotype, which I already worked out, we've written also in the table. So what are the available gametes? Nice and straightforward in this situation. The male is capital G, capital N. The female is lowercase g, lowercase n. And obviously when you do that first cross, it does indeed mean that all our individuals will have the genotype capital G, lowercase g, capital N, lowercase n. And if we have a look at what that means in terms of phenotype, remember that dominant gene produces the grey body and that dominant wing type produces the long wing, and 100% of the offspring will look like that. So now we're going to cross that F1 generation that we've just produced. So we're going to have two parents with the same phenotype, grey long wing. As we know, the genotype for both will be capital G, lowercase g, uppercase N, lowercase n. And this is the crucial difference between when we did the dihybrid cross versus now looking at autosomal linkage. Look, we only have two combinations of potential gametes as opposed to the four that we saw in the dihybrid cross. And why is that? Well, it's because of autosomal linkage, and it's because those genes are found on the same chromosome. So it means that there's only two gametes. So look for the wording in the exam question. If it says the genes are unlinked, then you need to use the example I used in my dihybrid cross video. If it says that they're linked, this is what you need to do. And then, as always, we're just doing a straightforward cross. Rather than that massive Punnett square from the previous video, it's a much more normal size Punnett square. Crossing the capital G, capital N, capital G, capital N means you're obviously going to have your homozygous dominant genotype producing your grey long wing fly. Same over here, but we've got the heterozygous genotype this time. Follows again that this fly will be heterozygous and be grey and longed wing. And then finally, it's only that fourth potential offspring that has the homozygous recessive genotype, meaning that you have black short wing flies. One thing they do like to point out in exams is that occasionally you get an anomalous result, occasionally you get a black long wing fly or a great short wing fly, and the reason for that is due to what I said before, the fact that crossing over has occurred in prophase 1 of meiosis. The first essential thing is that we look at the definition of epistasis. So I've stated that here. Epistasis is when the expression of one gene masks or affects the expression of another. And what we're saying here is that the phenotype of some alleles is able to directly alter or mask the phenotype of a second gene's alleles. Remember for me that a phenotype is the physical appearance of a gene. That could be eye colour, fur colour, whether you have a particular disease such as cystic fibrosis. Remember that an allele is simply a form of a gene. So if you're looking at eye colour, for example, this would be the allele for brown eyes, this would be the allele for blue eyes. We're going to jump straight in by looking at an example. So there are two genes in mice that control fur colour. Genotypes uppercase A, or uppercase A, lowercase a, have black bands in their fur. So we're saying that these mice have black bands in their fur, which we call agouti. The lowercase a situation, so the homozygous recessive situation, means that they have solid black fur. So first of all, be aware that if you have capital A, lowercase a, you're going to have black banded fur. If you have the homozygous recessive lowercase a situation, you're going to have solid black fur. We're then told that gene B controls the expression of gene A. So whether the mice have black banded fur or solid black fur will be dictated by gene B. So the genotypes uppercase B, or uppercase B, lowercase b will allow the expression of gene A, but if you have that homozygous recessive situation, you will not get the expression of gene A. And as a result, mice with the genotype lowercase b will be white, and we call that albino. So if we look at some pictures to understand what's going on, first of all we need to look at what's going on with gene B, because when we identify what's going on with gene B, we'll know what output of gene A we'll get. So we know that capital B, or capital B, lowercase b will allow that expression of gene A. So we've got that situation there, which means we get the expression of gene A, and we've been told here that if we allow that expression of gene A, then the mice are going to have black bands in their fur, and that's called agouti. I know it's a bit confusing because it doesn't look very black, but we have our agouti mouse here. Now with gene B, because again we've been told here these two genotypes will allow that expression of gene A, so we're allowing the expression of gene A, which in this situation is lowercase a. That means that we have a mouse with solid black fur as shown here. Lastly, because we've got lowercase b, we will not allow the expression of gene A. So it doesn't actually matter whether that mice was supposed to have, in the case here, agouti fur colouring, or in the case here, solid black. Quite simply, neither of these genotypes will be expressed, and that's because that lowercase b has masked the expression of those genotypes, and that's why we have our albino mouse. Now let's look at a cross. So we're looking at two offspring of an agouti AABB mouse, and we're crossing it with an albino mouse. So let's look at the situation. We know that we needed either a big B, small b, or big B in order to allow the expression of the agouti genotype. So we've got our agouti mouse there. Obviously all the gametes, sex cells, produced will be capital A, capital B. We have our white mouse, and remember the only genotype which produces a white mouse will have lowercase a, lowercase b, because remember that will mask the A allele. We know that all our gametes are therefore going to be small a, small b. So fundamentally our F1 phenotype, the first lot of offspring, must all be agouti. Why? Because they have the capital B, lowercase b, which means we express the capital A, lowercase a, which remember, looking at above from our key, is the agouti colouring. It now gets more complicated if we look at the gamete combination produced. Remember that these genes are unlinked. We haven't been told that they're linked, which means we get this combination where you can have one of 4 gamete combinations. Look at another video if you're unsure as to how I've done this. But now we're ready for our giant Punnett square in order to do the genetic cross. Just to remind you, if you have either capital B, lowercase b, or capital B, then no masking occurs. So if you have either capital A, or capital A, lowercase a, you're going to have an agouti mouse. What about if we had the homozygous recessive genotype with the capital B, lowercase b, or capital B, then you'd have your solid black mouse. Let's look at the final combination. So if we have that homozygous recessive genotype, we'd be masking our A gene. So it doesn't actually matter what combination of A's you had, you'd always end up with albino. And let's just make sure that checks out as we look at the table. So in our first cross, AB by AB, we end up with capital A, capital B. So let's just double check. There's our capital B. There's our capital A, and look, we have a Guti, which is shown by that tan colour. Moving on, capital A, capital B, lowercase b. That's that situation. Again, pushing out a Guti. Now let's double check this one. Capital A, lowercase b. So we're starting here, lowercase b, capital A, look, albino, right, it's white, it is albino. Let's check one of the black ones. So lowercase a, capital B. There's your capital B. There's your lowercase a. Look, solid black, it works. So let's look at our phenotype ratio. We have 9:3:4, agouti, black, albino. And you must be prepared to list all the different genotypes which account for them. So for example, for agouti, you can just pull them out of the table. So I'm just going along this top row here. I'm not going to do them all, it would take all day, but let's have a look at our blacks. So that's these three here. Let's look at our albinos. So it's these four here. So notice a 9:4:3 ratio is typical of a recessive epistasis. Remember, it is recessive because remember, it was only when we had this combination here that you mask that A gene. So let's first of all look at what the chi-squared test is. So it's a statistical test used to establish whether the difference between observed and expected results is small enough to occur purely due to chance. Notice it has to be used where we have a particular outcome that we expect and we have a list of observations and we're looking to see if those observations are different from the expected results due to random chance or because there is a significant difference. So it can be used to test what's known as the null hypothesis. A null hypothesis is one in which the results of a scientific investigation will produce no statistical significance. For example, there is no difference in the number of times a flipped coin will land on heads and the number of times it will land on tails. So we'll take the flipping a coin example because I think this gives a really nice clear image of how the chi-squared can be used. So if a coin is flipped 100 times, it would be normal to assume that it would land on heads 50 times and tails 50 times. But in practice, we know that it's unusual for these exact results to occur. And so if it lands on heads 55 times and tails 45 times, if you toss it 100 times, is this because the coin was biased or is it purely down to chance? And the chi-squared test will help us determine which explanation is correct. So carrying out a chi-squared test, the chi-squared formula looks like this. We have chi-squared is the sum, which is this symbol here, of the observed value minus the expected value squared divided by the expected value. And if we use the table like this, we can see how we're going to use that formula. So going back to our flipping a coin example, we know from the information on the previous slide that the observed number of heads was 55. The expected number was 50. So if we do observed value minus expected value, we get a value of 5. If we square that, we get a value of 25. And then if we do that number divided by expected value, which remember was 50, we get 0.5 as our answer. Now looking along the tails row, we know from the previous slide that our observed number was 45. We expected there to be 50. So if we do observed minus expected, we get minus 5. If you square a negative number, it becomes positive. So minus 5 squared is 25. If we do 25 divided by our expected value, which was 50, again, we get 0.5. We sum together those two numbers to get an answer of 1. And so now we've calculated our chi-squared value, we need to compare it to a chi-squared table. And in order to do this, our first step is to calculate the degrees of freedom. Now this sounds complicated, but I promise it's quite straightforward. You just need to know what the degree of freedom means, and all it means is you need to take your number of categories. In the case of the flipped coin, that's heads or tails, and then you need to minus 1 from that answer. So because we have 2 categories, either heads or tails, we minus 1, therefore our degree of freedom is 1. And the importance of the degrees of freedom is it tells you which row to look at in the table. So because we've calculated 1 as our degree of freedom, we know we're looking along this first row. Then go back, have a look at our calculated chi-squared value, which remember was 1. And we're looking to see where that 1 falls along that first row. And as the arrow points out, it falls here. Now the chi-squared table, as you can see, is split into 2. You've got this larger box here followed by this box. Now notice if your calculated value falls into this larger box on the left hand side, we can see that our results are not statistically significant. If our value calculated had fallen into this box over here, then we would say that our results are statistically significant. So the location of your chi-squared value will give you a critical value known as P. And the critical value is the probability that chance alone could have produced the results observed. So in this example, our critical value, being 1, fell between these 2 columns here, as 1 sits between 0.455 and 1.32. And effectively, because we know that it's not significant, all we're saying is that that calculated chi-squared means that the probability that our results were due to chance was between 25% and 50%. And that's what these numbers here tell us. So 1 falling in this position here tells us that the probability that our results were due to chance stands between 25 and 50%. And so if we look at this slide, because our p-value was between 0.5 and 0.25, we know that our results were not significant, and so therefore the null hypothesis must be accepted. So just to write it out, we're saying that differences in the number of tails and heads produced are due to chance only, i.e. the coin is not biased, and therefore we're saying the null hypothesis is accepted. And so you need to get used to using the right terminology. So we've already discussed our example here, but let's back up a little bit and look at our general overview. So we know that the critical value P tells you if your results are significant. For results to be significant and the null hypothesis rejected, the probability that observed results were due to chance must be less than 5%, and therefore the p-value must be less than 0.05. So if we have a look, all that's saying is our chi-squared value, based on the degrees of freedom which we would have calculated, must fall somewhere in here for the results to be statistically significant. If we'd had more categories, then obviously we'd be looking at a different row because we'd have a differing degree of freedom. So our criteria for carrying out a chi-squared test: firstly, the sample size must be large, so it must be over 20. It must be used for data that falls into discrete categories, so discontinuous data such as heads or tails, eye colour, fur colour, and only raw counts must be used, not percentages or rates. Before we get into the nitty-gritty of the Hardy-Weinberg Principle, let's first of all look at a few key definitions. The first one is gene pool. Now the gene pool is all the alleles of all the genes of all the individuals in a population at any particular time. The allelic frequency now is the number of times an allele occurs within the gene pool. And as always, it's so much easier to understand what these definitions mean if we look at a particular case study, and our case study today will be examining the disease cystic fibrosis. So just to remind you, remember cystic fibrosis is caused by a recessive allele, and so if we use an F to represent that allele, you'll need 2 copies of that lowercase f in order to get the disease. If you have either uppercase F, lowercase f, or 2 uppercase Fs, then your person will be healthy. Although obviously this genotype here will mean that you're a carrier of the disease. Cystic fibrosis is a disorder where excess mucus is produced. So let's work out how we can use cystic fibrosis to help us understand the meanings of the word gene pool and allelic frequency. So we're going to start by making the statement that any individual has 2 of these alleles in every one of their cells. And so if there were 10,000 people in a population, then there will be 20,000 alleles in the gene pool for this gene. So if we look at the 3 possible genotypes, which I've already mentioned, and these are the genotypes that relate to cystic fibrosis. The first one is the homozygous dominant genotype. Homozygous meaning the same allele, dominant meaning that it's the capitalized version. You have the homozygous recessive genotype. Homozygous meaning, again, 2 of the same alleles, recessive meaning that they're both lowercase. And then the third type is the heterozygous version, which remember could either be uppercase, lowercase f, or lowercase f, uppercase f. It could come in either order, so do be aware of that. So if we look at the Hardy-Weinberg Principle, the first thing to be aware of is that in any population, the total number of alleles is taken to be 1. So that's just a statement you need to be aware of. So it's a bit like the probability topic in maths, where 1 is 100%. Now let's, for argument's sake, say that within a particular population, pretend that the genotype capital F was true for every single person in the population, then we would say that the frequency of that dominant allele F would be 1. And therefore it makes total sense to say that the frequency of the recessive allele lowercase f would be 0. How about we take a different scenario now, which is that we say that everyone in the particular population was heterozygous, so therefore had an uppercase F, lowercase f, and then we could make the following statement. The frequency of the dominant allele F would be 0.5, and it follows that the frequency of the recessive allele F would be 0.5. So what is the point of the Hardy-Weinberg Principle? Well, it's used to calculate the frequencies of the alleles of a particular gene in a population. The Hardy-Weinberg Principle also predicts that the proportion of dominant and recessive alleles of any gene in a population remains the same from one generation to the next. However, this is only true if the following is adhered to. Number 1, no mutations arise. Number 2, that the population is isolated. 3, that there is no selection pressure. 4, that our population is large. And 5, that mating occurs randomly. So now we're actually ready to look at the Hardy-Weinberg Principle and what the equation for it actually is. So notice that the dominant allele is given the letter P, whereas the recessive allele is given the letter Q. Because we said that all the alleles in the population must add up to 1, it therefore makes sense that P plus Q equals 1. Let's use our cystic fibrosis example here, just to prove to you that it all makes sense. Remember that dominant allele in my example above was F, and my recessive allele was lowercase f, and therefore F plus F equals 1. Now remember that those alleles come in pairs, so we have a potential combination of these genotypes within one particular population. And again, we know that that will add up to 1. Because in the Hardy-Weinberg Principle we use P to represent capital F, because it is the dominant allele, we can therefore write this. And remember that we use Q to represent our recessive allele. Let's gather together our terms. So P times P, remember, is the same as P squared. We've got 2PQ if we add PQ and QP together, and then we have Q squared. And these two equations are the important things to be aware of with the Hardy-Weinberg Principle, and they're the two equations you're actually going to use to help solve a question. Everything else in pink is just me showing how we derive this. So let's look at some past exam practice. Lyra Shush. Sea otters were close to extinction at the start of the 20th century. Following a ban on hunting sea otters, the size of their populations began to increase. Scientists studied the frequency of two alleles of a gene in one population of sea otters. The dominant allele T codes for an enzyme. The other allele, lowercase t, is recessive and does not produce a functional enzyme. In a population of sea otters, the allele frequency for the recessive allele T was found to be 0.2. Use the Hardy-Weinberg equation to calculate the percentage of homozygous recessive sea otters in this population. Show you're working. So the first thing we want to do is just write out the two Hardy-Weinberg equations. It's always really good practice to do this. And now let's make some labels as to what we know from the question. So we know that the allele frequency for the recessive allele T was found to be 0.2. Remember that the recessive allele is Q in this situation. So we know that Q equals 0.2. Look back at the question, what's it actually asking us? It's asking us to calculate the percentage of homozygous recessive sea otters. Well, the homozygous recessive ones are therefore by definition Q squared. So you simply do 0.2 squared. And then the last place to make sure you don't slip up on is the fact that it needs to be a percentage. So times by 100 to turn that into a percent. So the answer here is 4%. What does the Hardy-Weinberg Principle predict about the frequency of the allele lowercase t after another 10 generations? It should remain the same, and that's because of everything else I stated about there being no selection pressures, being a large population, random mating, etc. What does the Hardy-Weinberg Principle predict? That's worth 3 marks. Remember that the Hardy-Weinberg Principle predicts that the frequency of alleles remains the same from one generation to the next. And that's providing that there's no selection pressure and that mating occurs randomly.